Hello, today we're going to go over DNA replication, and this process is a very complicated process, but we're going to take it slow and make sure we get all the pieces in. So when we're talking about the replication of DNA, this is something that Watson and Crick kind of hypothesized when they came up with their model of DNA, obviously with the help of Rosalind Franklin. And they noticed that the way that the bases paired seemed to indicate that perhaps one strand could serve as a template for a new strand. And this is indeed how they discovered and began to discover that DNA was copied. So there was a few different models of the way DNA could be copied. One was the conservative model, in which there was one parental strand, and that parental strand was copied, and the two parental strands would come back together after it was copied. So that way you would have one totally original strand, one totally new strand, and then after a second replication, you'd end up with one totally original strand and three totally new strands. The dispersive model was a little bit even more confusing, in which all four strands of DNA after the second round of replication have mixtures of the old and new DNA. So this is a little bit more complicated to think about how this might actually have worked, but it nonetheless was an experimental prediction. And then finally, we have this option, which was known as the semi-conservative replication, in which after one round you have one strand of old parental DNA, one strand of new DNA, and both. And after both rounds, you end up with one strand of the original and one strand of the new. And then two of those end up with two brand new daughter molecules of DNA. And we come to find out that this is actually the model that our DNA follows during replication. So this is kind of a little zoom in on what the semi-conservative type of replication actually looks like, in which we end up our daughter or parent strands separate, a new daughter strand forms in each one, and then we end up with half of the old strand and half of a brand new strand that's complementary to the old strand. So basically, when with the semi-conservative model, since the two strands of DNA are complementary, each strand, when they separate, can act as a template for building a new strand. So the parent molecule will unwind, and the two new daughter strands are built based on those base pairing rules that we talked about in 13.1. So for example, A pairs with T, G pairs with C, and vice versa. So they're able to build the new strand in that way. This was originally hypothesized by Watson and Crick, and they believed that when the DNA molecule replicated, it would have one old strand and one new strand, hence the semi-conservative model. And as we mentioned, we kind of already discussed some of the old other ways that people thought this could have happened. And this is just another picture to kind of show you how this worked. There were two other guys named Mendelssohn and Stahl, and they did an experiment to prove this. So what they did was they cultured the bacteria in a medium with an isotope of nitrogen 15, very heavy isotope, and then they transferred those bacteria to a medium with nitrogen 14, the lighter isotope. They centrifuged the DNA, so they spun it really fast to separate it out after its first replication, and then they did the same thing after a second replication. And what they observed was the more dense isotope was present in both the first and the second replication. That was our original, if you remember. And then when it had gone through the second replication, it also exhibited some of the new DNA, which was the lighter isotope. So that was how they were able to prove the semi-conservative model, because if it was conservative and the parent showed up in both, or as it was, they would have seen a band. They would have seen a full band in both. With semi-conservative, they should have expected to see the heavy band in one, and then, or partially heavy band in one, and that would be both of these. And then in this new one, they would see the partially heavy band and then the lighter band from the new DNA. And if it had been dispersive, they probably would have seen all kinds of stuff. And what they ended up seeing was the first round of replication produced kind of a medium density band, which indicated there might be some N15 from the original sample and N14 from the new sample. And then in the second round of replication, they had a very light density band, which indicated those two brand new daughter DNA molecules, and then the middle density band, which indicated the bands with the one parent strand that contained the nitrogen 15. So when we take a look at DNA, a couple of things to note just in general is that it is very impressive in how quickly and accurately our DNA can be copied. It's really, really a miracle, so hopefully you appreciate it for the miracle that it is. Also, there are tons of different enzymes and other proteins that participate in DNA replication. If you read the textbook, you're going to see a lot of enzymes that are listed, but I've tried to isolate the ones that you need to know for the AP exam for this particular presentation. We also know a lot more about how DNA is replicated in bacteria, and so we'll be using E. coli bacteria as a model, but a similar process also happens in eukaryotes, and for the most part we've come to understand that the process is very similar between both prokaryotes and eukaryotes. So if we continue on, you will see that the replication actually begins at certain sites called origins of replication. And at these sites, you know, DNA is a double helix with two strands. Those strands get separated and they form what's known as a replication bubble. And then at the end of each bubble is known as a replication fork, and that's kind of the Y-shaped region where the parental strands of DNA are being unwound. So an enzyme helps catalyze this, and helicase is actually the enzyme that really unwinds our DNA strand, and you can see this bubble would form, and then where this Y is, that's known as the replication fork. And then topoisomerase, this is an enzyme that is important because since the DNA double helix is typically twisted, if we were to try to untwist it, you can imagine it would cause a lot of strain. So the topoisomerase kind of helps relieve some of this strain by breaking, swiveling, and rejoining the parental DNA ahead of the replication fork. So kind of preventing that strain that might come along. So this is kind of the same thing. The helicase is the one that untwists the double helix at the replication fork. Single strand binding proteins bind and stabilize the single stranded DNA, and then once again topoisomerase is used to help relieve that strain, topoisomerase. And that's very helpful as well. So these are all enzymes that help with the replication of DNA really before any of the replication begins. In E. coli cells, as well as other bacteria, the DNA is found in a circular form. So typically, since it's circular, the replication fork kind of starts and literally forms a bubble within the circular DNA. In contrast, our DNA and those of other eukaryotic organisms is linear DNA. So there can be multiple replication forks in eukaryotic DNA in order to have multiple replication experiences happening at the same time, as opposed to in bacterial DNA, typically there's only one replication bubble, a replication fork, and it replicates fairly quickly. However, the linear DNA of the eukaryotic cell has many origins of replication. It could have up to hundreds or even thousands, and whereas the bacterial just kind of goes around the circle. So I'm going to stop the video here because now we're going to end up getting to an even more important part of how DNA is replicated and how that actually happens. So that will be in the second part of this video.